We're in the second part of vision. I'm going to start out by going over transduction, so repeating that for one of my classes. Then go over the concept of lateral inhibition, starting with the neuron's receptive field and how this inhibition is part of the communication across the retina. And then I'll just start talking about color vision in this section, and then I'll have to finish it in the next section. I'm going to start with the transduction and how that happens. I kind of bumbled over this with one of my classes, and then we didn't get to it in the other class, so I'm just going to repeat this slide for one of you groups. So we're talking about what's happening in the photoreceptors, and we can sing it. This is where transduction. So a neuron's receptive field is the part of the visual field that either excites or inhibits a particular cell. And for now, I'm talking about the visual system of the brain, but every sensory system is going to have those neurons are going to have their own receptive field of what they're responding to. And I think I mentioned in my Section 3 class how important this is to know if you're doing something like physical therapy or massage therapy, that where you're touching a person, that we're going to see these same kind of center surround receptive fields in that somatosensory system. But we're going to talk about it for now with the visual system. And just note that whether I'm having an excitatory effect or an inhibitory effect, and if I'm influencing a neuron, I'm part of that neuron's receptive field, where really what we're talking about is the visual field, so the receptors that are responding to some part of the visual field, and then communicating on to neurons. So those neurons, the receptive fields have different shapes and sizes and properties, and we see that they are larger when there's more convergence. So if I'm getting more information from more neurons, my receptive field is going to be larger necessarily, and we're also going to watch them get larger and more complex as we get farther and farther from the retina. So one example, we're going to start with this center surround receptive field. As we talk about how we measure the receptive fields of neurons, this is how we how people originally did this in various areas of the brain, but I'm going to start here recording from a single fiber of the optic nerve. Remember that these are the axons of the retinal ganglion cells that are in the eye. And just so I kind of remind you of that, we have this upside down and reversed image. So the light is being reflected into our eyes in such a way that it's hitting the back of the eyes. It's upside down. That image is upside down and reversed. And so the way they did this was they would have a cat and they would anesthetize the cat, anesthetize so that the eye was still, and then they would show things on a screen. And so if I'm showing something at the top of the screen at A, that's going to hit the bottom of the retina down at A prime. If I show something in the center at B, it's going to hit at B prime. Show something down in the lower part of the visual field at C, it's going to hit at the top of the retina at C prime. And so usually I do this trying to show my light on these different parts, but if you look at the images over on the left, and at the very left we see what the cat is looking at on the screen. And then what they would do is show the light or whatever stimulus they had on A. And if they were showing this stimulus there, it was a circular stimulus. A, we see the firing of the neuron over in the right of that picture. That those figures are showing the firing of neurons, and you can see that the firing is at the spontaneous rate. So everything in A, regardless of where they were on the kind of outer part of that screen, was not part of that neuron's receptive field. Okay, again, they're recording from a single fiber in the optic nerve, or a single retinal ganglion cell. If they shine the light somewhere in B, so in that center that has a little plus in it, you can see over at the figures, at the graphs, that when that light came on, at B, the neuron started firing, and when the light went off, the neuron stopped firing. So that's an excitatory part of the receptive field. If they shined the light somewhere in C, so with the negatives in it and the surrounding B, what they found was that that neuron, when the light came on, the neuron would stop firing. When the light went off, we would see this kind of rebound effect. So this is showing that when we turn off the spontaneous firing of a neuron with inhibitory information, and then we take that stimulus away, we see a bit of a rebound where the neuron fires. And so this is an example of a center surround receptive field, excitatory center, inhibitory surround, but it looks like we have just as many inhibitory center, excitatory surround receptive fields as well. And I've taken this picture over at the right from Laura Freeberg because I like how she shows that this is the receptive field is really talking about the receptors that are, so the receptors that are responding to the visual field, their influence on a neuron. In this case, looking at a bipolar cell, which is going to communicate to my retinal ganglion cells, we're seeing those center receptors that are purple. She's showing that has an excitatory effect in this case, and those yellow receptors that are showing that they're having an inhibitory effect. So she's just coloring the receptors to show what kind of effect they're having, but they're all just rods in this case. And as I mentioned, if there's more convergence, we're going to have larger receptive fields. Receptive fields are going to become larger and more complex the farther we get from the retina. So right now we're talking about the retinal ganglion cells having the center surround receptive fields. And I didn't really read what was at the top of the screen on the last slide, but we're going to see those same center surround receptive fields in the lateral geniculate nucleus. So that is the very first structure that we see the retinal ganglion cells are going to synapse in the lateral geniculate nucleus as well as the superior colliculus. But we're going to get, as we move away and we get into higher level processing, those are going to become more complex than just little dots. And so sometimes I use this example of the frogs what they're seeing basically with those subcortical structures and not having the cortex that we have, they're seeing the world of little dots. And so their food is going to be, their fly is going to be a little dot. And they see that pretty well. They can see, so they can put it together into kind of shadows. So if there's a predator, coming over. They can kind of overlay those, what they're seeing is a bunch of little dots, but overlaying each other. And then what they see the very best, what their visual system responds to the very best, is a little dot that is fly-like and moving around in a fly-like kind of way, so that they can, so that they are evolved to obtain their food. We get to put this into much more complex receptive fields as we get later into. We get to pull this apart and put this back together into features from little dots, and then put it back together into tables and chairs and trees and dogs and people and faces and all of that. So there's no good direction to talk about receptive fields versus lateral inhibition, because lateral inhibition is helping us with that center surround receptive field, with the inhibitory part of that receptive field. But either way, you have to talk about one before the other, without the one making complete sense without the other. So if you want to, sometimes I think it's good to go back and start it all over after hearing about lateral inhibition. So this, on the left-hand side, those words are really repeating, except we're adding just a little tiny bit, because we've talked about the photoreceptors, that we have 126 million per eye. They're going to communicate through the bipolar cells. We're going to see some convergence here, where many photoreceptors are going to communicate to a single bipolar cell, because when we then converge onto the retinal ganglion cells, we only have 1 million retinal ganglion cells that are sending their axons to the brain, as they are those axons making up the optic nerve. So that is repeating, and you can see the pictures over to the right that are showing about 8 or 9 or 10 receptors communicating to 4 retinal ganglion cells, and why we need this kind of convergence. But remember, 126 million photoreceptors need to communicate to 1 million retinal ganglion cells. So we have a bunch of convergence. If you remember, that's mostly in the rods, but also in the cones that are in the periphery of the eye. That kind of convergence is not happening in the fovea. But we've added some cells, the horizontal and the amacrine cells, and we've added the words lateral inhibition, because really, so if you look at the picture to the right, the horizontal cells are going kind of perpendicular to the basic communication that's happening, and so are the amacrine cells. And so they are allowing, by going perpendicular to those cells, they are communicating laterally, as it's called, and oftentimes they are providing inhibition, some amount of inhibition from our excited neighbors. Okay, so usually I draw a picture and I talk through it, and if we need to do that on the next Monday, if people really want to know how lateral inhibition works to allow us to see contrasts and changes in our environment, we can do that. I think it's an important thing to understand, but I tell students, because a lot of students aren't going to understand this, if you just memorize this paragraph, or realize that if I ask a question about lateral inhibition, I'm going to give you basically a paragraph very similar to this, and you're going to fill in the blank with lateral inhibition. Okay, lateral inhibition refers to the fact that an excited neuron may reduce the activity of its neighbors, which means that if a single neuron is active, it responds more strongly than if its neighbors are also stimulated, because they're often sending inhibition to neurons along the same path. And what this really means is if a single neuron is active, then the neuron that it's communicating to, that can respond more strongly than if its neighbors are also stimulated, because those neighbors can be sending inhibition. And lateral inhibition is the retina's way of sharpening contrasts to emphasize the borders of objects. And I often say when I'm talking about lateral inhibition, a sensory system has got to work this way, regardless of what planet I'm on or evolved on, what sensory system it is. We want to hear relative information. We want to see and feel relative information, that we know where things are changing. We don't want absolute amounts of light. Light upon light upon light would be overwhelming, and it would be too much information, and we wouldn't see the changes in the environment, the object, in comparison to the context or background that it's in, but I want to see where the table starts and where the table ends so that I don't run into it when I walk, so that I put my cup on the table and not somewhere in between the table and the floor so that it falls to the ground. But this is how sensory systems have to work. I'm going to go through this explanation of an illusion to kind of show you that lateral inhibition is happening. And if you're in my sensation and perception class, you know that it's a little bit more complicated than this, and we have some illusions that we cannot explain with lateral inhibition, even though the same kind of thing is either happening, but it's happening in a different way. But just so we're doing this one time, this simultaneous contrast is an illusion. And if I ask you, when you're looking at this screen, at the center squares, whether the one on the left or the one on the right appears to be darker, some of you know the punchline, that they are in fact the same exact shade of gray, the same exact amount of light is being reflected into your eyes, but most of us perceive the one on the left as being darker than the one on the right. And I'm going to explain this with my next picture. With the one on the left appearing darker, it's in a lighter background, and that lighter background is sending more inhibition, so that appears to be darker in comparison to the one on the right, because the one on the right is in a dark background, and because the background is dark, that is sending less inhibition to that square. So that one appears lighter than the one on the left. Okay, again, I'll suggest that if you want me to go more through lateral inhibition, I can. And if you just want to go, maybe repeat what was just said so that the receptive field of a neuron, you can listen to it after learning about lateral inhibition and then go back through. Either way, it's a pretty complicated concept, but again, it's really the way sensory systems have to work. We're going to move on to the theories of color perception. And we're going to talk about, I think in this class we talk about 3 different theories and how they each are explaining some aspect of color perception. The first one is usually called trichromatic theory of color vision. Sometimes it's called the Young-Helmholtz theory of color vision. You can see that Thomas Young is one of the people who told us about trichromatic color vision. Hermann von Helmholtz came along quite a bit later, and he had a much more respect, was much more well known than Young, and he wrote it up and did some clear description of how it works. They both are saying that there are 3 basic color mechanisms, and this is how we get the name trichromatic, right? Tri, 3, chroma, color, 3 color mechanisms. And they learned this using behavioral evidence, because if you know, back then in the 1800s, we didn't have this kind of way of looking at the neurons of animals, or these kind of sophisticated ways of looking at brain imaging or any of this. So they were using behavioral evidence. They discovered this through using metamers, where metamers are things that are in the world, are physically different from each other, but when they go through our nervous system and we perceive them as being identical. So this behavioral support back in the 1800s came from color matching experiments, where they would start with a test field. On the left, you can see that this one is an example of 500 nanometers, a monochromatic light that is 500 nanometers wavelength. But that test field they used several different wavelengths. This is just one example. And in the comparison field, they would have the participant try to adjust the light levels in the comparison field so that it would match the test field, so that we would get these metamers. Okay, if they gave the person one wavelength, so I'm going to just start with the 420 nanometers. I start out with 420 nanometers. No matter how much I adjust that, I cannot match the 500 nanometer light. Okay, they give people 2, so 420 and 640 nanometers, those 2 lights, and they can adjust them as much as they want until they can match something in the test field. In this case, they could match some, of the lights in the test field, but not all of the lights. There were always some lights that they were not able to match when they gave them 3 different wavelengths. So in this example, 420, 560, and 640 nanometer lights, they could adjust those 3 lights such that they could match any color or wavelength that was in the test field. They needed 3 to match any. They never needed 4 to match some color in the test field. They couldn't match all of the colors, all of the possible single wavelength lights in the test field with just 2. They needed 3, which told them, Young and Helmholtz, that we had 3 color mechanisms. Or this was the assumption from the behavioral evidence. It was really about 100 years after Helmholtz that we obtained the physiological support. So I want to say this study was done in 1957, if I'm remembering correctly, where they measured the absorption of single receptor cells. So the reflectance of light, they used the reflectance of light and looked at the absorption of single receptor cells. And what they discovered was these 3 cone types, right? Short wavelength cones that responded maximally at 419 nanometers, medium wavelength cones that responded maximally at, or absorbed maximally at 531 nanometers, and long wavelength cones that absorbed maximally at 558 nanometers. So 3 cone types, 3 color mechanisms. Are 3 color mechanisms necessary for color vision? Well, yes, for our range of colors, but no, we could see with just 2 of these cones or 2 color mechanisms. We just wouldn't see the same range of colors. And so we've already started some of this discussion in my section 1 class because they were asking questions about color blindness, or what's better referred to as color deficiency, because they are deficient in their color vision. When we are talking about inherited color deficits, what's happening there is that we have those photopigments, if you remember, that are making up the different photopigments in rods and different photopigments in the different kinds of cones, that for some of us, those are either defective or absent. And there are a number of different kinds of color deficiency. We determine a person's color vision abilities by using these color vision tests. One is the Ishihara plates that I'm showing here. If I ask you what you see on the left, most people tell me they see a 74, right? This green 74 in the red background. And if I ask what you see on the right, it looks like maybe this lighter green colors, and people think, is that a 21? Is that a 24? I'm not really sure what it is. It's really difficult to see. Well, I've had several, so at this point I think I've had maybe 7 or 8 people who are color deficient in my various classes, and they assure me that the one on the left to them appears exactly like the one on the right. They cannot see the 74, and what they do see is what we see on the right, where there's like, it's really hard to see. Is it a 21? I'm not really sure. Okay, and that's how we test for color deficiency. We can also test for color deficiency by using those kind of color matching studies that I was talking about how they learned about trichromatic color vision, that we have these 3 mechanisms. But again, those people who only have 2 of the photopigments and are lacking some range of colors, they can match any monochromatic light in the test field with only 2 lights in the comparison field. Now, if we were standing behind them, we'd say, hey, that doesn't match, but to them, it matches. So the photopigments are defective or absent. Mostly we see this in dichromats. That's more common, where they're missing a single photopigment. This is something that is on the X chromosome, so it's one of those sex-linked genes, and females are less likely to become color deficient. They need both their mother to be a carrier and their father to be color deficient for them to become color deficient, and they need to get those 2 X's that are showing this recessive trait. whereas men only need the one from their mother because they have the Y chromosome from their father. That's not, so they're just going to show that recessive trait if they receive that genetically. Monochromats, we see 10 out of 1 million people. So again, this is very rare. They are missing 2 photopigments or missing their cones. They're sensitive to bright lights. They asked me about this in the section 1 class, and I guess I spaced what was on my own slides and hadn't really thought through it enough for it to stick with me. That yes, they do have poor visual acuity, so it is kind of blurry because they don't have those tightly packed cones with their one-to-one wiring to the brain. Their vision is based on the rod system, so they're going to be better off at night, and they're going to have a hard time in sunlight. So I'm going to walk through a few of the color deficiencies that we can see, and I'm just going to go ahead and say that the picture on the right is what we are assuming they see. From what I understand, for the people who are color deficient in my classes, these 2 pictures do appear about pretty much the same to them, but we don't really know if they're seeing that as green. And while I think a lot of them have learned to point to those colors and say they are green, I have had one female who was color deficient in my class, and she said that those colors looked kind of peach to her, and they all looked kind of different shades of peach. So I don't know if it's what we've learned to call colors, but we don't really know what they're seeing, and they don't really know what we are seeing. But I'm going to start with this protanopia, where the long wavelength cone is defective or absent. This happens in about 1% of males, about 0.02% of females, and you can see that they're missing the range. They're not really getting the differences between the reds, yellows, and greens, those in the medium to longer wavelengths. Deuteranopia is when the medium wavelength cone is either defective or absent. This is in about 1% of males, about 0.01% of females. And in my classes, I usually go back and forth between these 2 to show that there doesn't appear to be much difference in the right hand, what's happening in the right hand image. But again, it's difficult to tell because we just point at colors and say this is what the color is, but what they're actually experiencing is not completely clear. And then we have tritanopia, where we assume that what's happening is a missing short wavelength cone. There are fewer people who have tritanopia. It's much more rare. And also there are fewer short wavelength cones. So this is the assumption of what's going on. This is happening in about 0.002% of males, 0.001% of females. So really quite rare. And you can see that now they're missing really the blue, indigo, violet end of the scale, and things are ending up looking kind of reddish, and they're missing some of that what's going to distinguish yellows from the reds and greens is going to be the short wavelength cone, and its little bit, whatever little bit of the ratio would have been short wavelength cones, they're not getting that information. And finally, we have monochromats, where you can see it looks very black and white vision. So they're missing 2 photopigments or all of their cones.